a marine biology master's student in the Schwederman Lab here at the University of Maine. And today I will be sharing a portion of my work on American lobster diet and thermal physiology, specifically the chapter of my thesis that focuses on the energetic cost of digestion. So to start with some background for this project, I don't have to tell anyone here that the Gulf of Maine is warming and experiencing increasing thermal variability and extreme thermal events. And as ectotherms, lobster distribution and physiology are profoundly impacted by temperature. They have a pretty broad thermal range, but start exhibiting signs of upper thermal stress when temperatures reach 20 degrees Celsius. And we know that living in an environment that's too warm has consequences like reduced settlement and increased susceptibility to disease, making this a primary concern for the lobster fishery. The fishery is also contending with changes in bait usage. Declining herring stocks have necessitated the use of alternative bait items like non-local fish, fish byproducts, and terrestrial items like cow and pig hide, things that lobsters would never encounter in the wild. And food impacts lobsters and all organisms' ability to deal with thermal challenges, which lobsters living in the Gulf of Maine are facing. And we don't have a thorough understanding of how these rapid changes in both food and temperature may be impacting lobsters on the sublethal level. And there are some physiological metrics that we can look at to try to elucidate the potential consequences of these changes. One of those is aerobic scope. Aerobic scope is the difference between an organism's standard metabolic rate, or SMR, and maximum metabolic rate, or MMR. And this represents how much aerobic activity an animal can do beyond just sitting there sustaining life. Things like moving, growing, reproducing. Generally, in ectotherms, scope is maximized at an optimal temperature, range of temperatures, and declines when it gets too hot or cold. These pinch points represent temperatures where aerobic scope is 0 and the animal has no capacity for additional aerobic activity. Eating food is an aerobic activity that takes up part of aerobic scope. Specific dynamic action, or SDA, is the rise in metabolic rate after eating a meal. And this rise represents the energy that's being used to digest, absorb, and assimilate that meal. And this process is illustrated by this figure. This is an SDA response in a lionfish, and we have time on the x-axis and oxygen consumption on the y-axis. So first, this black dotted line represents SMR, or baseline. The lionfish eats the meal, and then oxygen consumption spikes up and reaches this SDA peak. Oxygen consumption then gradually returns from this peak back down to SMR over the course of the SDA duration. And then the total area, the shaded area under the curve, is just called SDA, or SDA total. As I mentioned before, SDA takes up some of an organism's aerobic scope. Postprandial residual aerobic scope, or PRAS, is how much scope is left over during the SDA response. When an organism is dedicating energy towards digesting a meal, it's diverting energy away from other processes. You may be experiencing this feeling right now. If you've ever felt like you need a nap after a big meal, it's because your body is using energy to handle that food and not do other things. The amount of scope taken up by SDA depends on a lot of things, but in general, in ectotherms, temperatures increase metabolic rates, and so we expect the SDA response to be larger at higher temperatures with a smaller PRAS. More protein also tends to result in a larger SDA response because protein levels in a meal are positively associated with protein synthesis, which is the construction of new proteins in the cell, and this is an energetically expensive process. Coming back to the fishery, I'm thinking about SDA and its implications in the context of lobsters eating bait in the trap, kind of as their last meal before entering the processing chain. Most lobsters are sold live and need to be in robust enough health to survive all of the stressors that occur on the way to market, which can take days or weeks. And having a larger PRAS may be helpful in that the lobsters have larger energy reserves to deal with those stressors, potentially increasing the amount of lobsters that actually make it to market. So it's valuable for us to understand how consumption of these novel bait items impact lobster physiology and if that impact is interacting with rising ocean temperatures. So my study aims to answer this question, specifically focusing on the novel bait items menhaden and pickhide, which were chosen in collaboration with Maine's Department of Marine Resources. This past summer, my technician, Alyssa, and I worked with the Lobster Institute to purchase lobsters from the South Bristol Fishermen's Co-op, and we measured their metabolic rates after acclimating them to either 15 or 18 degrees Celsius for at least 3 weeks and feeding them one of these novel baits to see if rates varied by bait and or acclimation temperature. We measured their aerobic scope and SDA using intermittent flow respirometry. For this presentation, I won't get into the details of how this worked. I just kind of wanted to show it because building this setup was really labor intensive and I wanted to give it some love. Here's what the setup looked like in real life. Oxygen is measured in each one of these little boxes, and each box contains 1 lobster. And the data I get from my oxygen measurements looks like this. So the red highlighted portions show the lobster breathing and decreasing oxygen in the chamber. And then we let fresh water into the chamber so oxygen levels go back up. And I can measure the rate of decrease in oxygen over time during those red highlighted periods and use that as a proxy for metabolic rate. So we went about measuring our metabolic rates by first establishing SMR by continuously measuring oxygen consumption overnight. And the next day, we elicited an SDA response to a meal by gavage feeding each lobster a slurry of one of 3 foods: blue mussels, menhaden, or pickhide. Blue mussel was selected as a 3rd food because it's a common prey item in the wild for lobsters. I don't know if this will play. It didn't play. Well, that was going to be an example of me and Alyssa gavage feeding a lobster, but we force fed it to them in a syringe and then measured oxygen consumption continuously for 72 hours to capture that SDA response. And finally, we chased all the lobsters using a standard chase procedure to get their maximum metabolic rate. And then for the results of all of that data collection, we did not see statistically significant differences for absolute aerobic scope, which is just MMR minus SMR, or for factorial aerobic scope, which is scope for increasing metabolic rate proportional to SMR. But looking at factorial scope, both temperature treatments for mussel and pickhide look pretty similar, but the menhaden treatment looks really different at 15 versus 18 degrees. For SDA peak, again, that highest post-meal metabolic rate that we recorded, also no statistically significant results, but the pickhide treatment was greater overall, and we can see that the 18-degree pickhide had a lot less variation in the data than the rest of the groups. The interactive effect of meal and acclimation temperature in the time that it took from feeding to reach that SDA peak approached significance, and we can see a really big difference in the distribution between temperature treatments in the menhaden and the pickhide, which was not present for lobsters fed mussels. The duration of the SDA response was not significantly impacted by treatment. We do see this kind of trend of duration getting shorter as we look from menhaden to mussels to pickhide. I'd also expect the 18-degree treatments to be shorter than the 15-degree ones, since metabolic rates are increased with increasing temperatures, but only sort of saw that for menhaden. For total SDA, or that shaded area under the curve, also no statistically significant differences between treatments. There was a significant interaction of meal and acclimation temperature and factorial rise. And factorial rise is SDA peak as a proportion of SMR. And we see this really clear pattern in the menhaden and mussel of factorial rise being higher at 18 degrees, but this pattern is absent in the pickhide treatment. And finally, postprandial residual aerobic scope, shown here as percentage of each lobster's aerobic scope remaining during SDA peak, also had significant interaction effects. And these data were really interesting considering the results of crude lipid analysis that were run on our samples by Hallie Bear, of the Hamlin lab here at Maine. Pig hide consisted of much more fat than the mussels and menhaden, and likely has a lot less protein. For reference, a standard piece of bacon has like half the amount of fat as our pig hide, and given such dramatic differences in nutrition, I was expecting to see clearer patterns between treatments. I'm interested if anyone in the audience sees something that I don't, maybe. So in conclusion, my study sought to contribute to our knowledge of lobster physiology by assessing the impact of the consumption of different bait items on SDA and aerobic scope in lobsters. And we didn't find a lot of statistical significance across treatments, but I did see some interesting patterns that I'm working on fleshing out as I continue to work on my thesis. It's possible that lobsters are generalist scavengers and therefore are equipped to handle a diversity of food sources, which may explain the robust responses to the different meals that we saw. However, a lack of response in the metrics that I measured doesn't mean that there aren't some other mechanisms that might be being compensated for and that need investigation. We need some further, more focused research in this area to get a more thorough picture of what's really going on when lobsters are eating these things to support a more sustainable and secure fishery for everyone who depends on it. Thank you to the Lobster Institute for hosting this event and everyone who helped me with this project. And I'll take questions or comments.
Thank you, Maddie. Does anyone have a question?
That's really interesting stuff, especially just the incredibly long time period it takes for these peaks to be reached and sort of come back off of. How does that interact with the biology in the wild, right? Where lobsters are most likely eating more than every 40 hours, right? So is this a peak that is something that would be seen in wild animals, or would they be riding that high forever, basically, just going from meal to meal, skipping off the top and never seeing us come down off of the top of that SDA, if that makes any sense at all?
The peak, that highest point, is very short-lived. It takes a while to return all the way back down to baseline, but they can eat another meal before they're all the way back down. I believe just foregut clearance time is a bit shorter than the time it takes to return all the way back down to baseline. So yeah, thank you.
Go here, then there, then there.
Hi, I just hoping you can clarify, were the treatments within individuals? So were you giving like the individual like all three treatments, or was it in like separate individuals getting separate treatments?
Each lobster was held at one temperature and fed one thing. So we didn't repeat. Yeah. Is that your question?
Yeah. Yeah.
Cool. Thank you.
Thank you. I was curious about the connection between metabolism and health. Is this something you've actually looked at, or is that in literature?
Something that I have looked at?
Yeah.
No. In the literature, I guess it confers health in that you having a larger aerobic scope allows you to do more things that would make you healthier, like get enough food to eat and escape predation.
Now,
Is that your
I was
question?
thinking of infectious disease.
I'm not sure. Maybe.
Okay, thank you.
Thanks for your question.
It's someone.
Thanks. I was wondering with the gavage feeding, where they're being fed a slurry, right?
Yes.
Would you expect them, if they were fed like the whole food instead, whether there'd be some differences as they have to then compete with breaking down the actual food totally?
Yeah. There's a lot of SDA encompasses like the mechanical breakdown as well as whatever happens on the inside of the lobster and post absorption processes. So yeah, we were not accounting for them like grabbing and chewing the food. We did have to force feed them a slurry because they would not eat the pig hide voluntarily. But all of the lobsters got the same treatment in that way. Yeah, good
Thanks.
question, thank you.
Very interesting. It reminds me of my PhD. I did all my PhD on shrimps and metabolic rates. So it's very, I was just wondering from what I noticed on lobsters, doing metabolic rates on stage 4 lobsters for a while, stress is very important. So how did you, for example, your lobsters that you force feed must be very stressed. So did you also have a set of lobsters that you would manipulate without feeding just to subtract, you know, the stress level? Because it's some of the lobsters that I measured were stressed after handling for like 48 hours.
Okay,
So
yeah.
it might impact your standard metabolic rate as well.
Yeah, we did not do a sham feeding. In my analysis, I did account, there's like a time lag that accounts for the stress of that handling. And that aligns with the amount of time that we saw after our exhaustive chase procedure. It took them to return back to their standard metabolic rates, but we did not do a sham feeding.
Okay. And
that-
probably your numbers, if you have time, if you do a PhD, maybe consider doing more, like more samples. The variability amongst individual is crazy when
Yeah.
it comes to metabolic rate.
Yes. Yeah, definitely. Thank you.
Anyone else? Back here?
Amazing. So very cool. So when you fed, did you standardize the amount of food or standardize like hopefully the amount of lipids or proteins that they were getting? How did you decide like volume?
I fed them a 2% of their body weight in each food. I was not able to get like an iso calorie feeding for each of them, but I fed them the same amount, like proportional to their body weight. Yeah.
Anyone else? We have time for another question. Yeah. Right. Sorry, Maddie.
Sorry, Maddie. These lobsters are in this container that's somewhat enclosed, assuming excreting whatever they've eaten as well. Is that affecting the O2 in the container? Like I know pig hide, for example, destroys water quality in a lot of systems. So did you see any of those like excrements affecting things, or was it not there long enough?
We saw a lot of excrement. As part of like standard respirometry procedure, you measure respiration, you measure oxygen consumption in the chambers with no animal before you do the test and then after you do the test. And you can account for like microbial respiration by that way, yes. So yeah, thank you.